Good afternoon, everyone. Thank you for joining us for our live webinar, which will be presented live by Craig Wheelock with the title Not Just Fat, Exploring Octadecanoids as a Novel Bioactive Lipids in Inflammation. My name is Maria Fedorova, and I will conduct the introduction and manage questions and answer session at the end. The webinar is recorded and will be uploaded on the Lipid Maps website and our YouTube channel within the next 48 hours. So the webinar will be around 45 minutes and we will have 10 minutes at the end for the questions. So please, you can post your questions and the Q&A function throughout the presentation. And to ask your questions at the end, I will ask you to unmute yourself. So today's webinar is being sponsored by Avanti and we are grateful for their ongoing support. Now I would like to introduce our speaker. Craig is the head of the Unit of Integrative Metabolomics in the Institute of Environmental Medicine at the Karolinska Institute in Sweden, where he serves as the director of the Integrative Molecular Phenotyping Laboratory. He is also a distinguished visiting professor of metabolomics at Gumna University in Japan. Following his postdoctoral work on lipid metabolism at the University of California, Davis, he conducted additional postdoc studies at the Keck Laboratory in Kyoto University in Japan. In 2006, he was awarded a Marie Curie Fellowship to relocate to Karolinska Institute. Research in his group focuses on molecular phenotyping of respiratory disease, with a particular area of interest in investigating the role of eicosanoids and other lipid mediators. The overall aim is to develop personalized molecular profiles that can be associated with individual lifestyles, environmental exposure, and acceptability to disease onset. He has been a founding board member of the Nordic Metabolomics Society from 2017 to 2021, a board member of the International Metabolomics Society, and currently serves on the European Respiratory Society Advocacy Council as the Director of Scientific Relations within the EU. Thank you very much, Craig, for joining us today, and please go on.
Great. Thank you very much. Appreciate the introduction and the invitation to be here. I'll start off actually by saying an apology to all of you on the call. I'm losing my voice a little bit, so it's kind of scratchy and weak, and I'll do my best to keep it going for the length of the seminar, but I will be drinking a lot. My apologies. So, as Maria said, I'll be speaking about my favorite topic, as you can see from the title here. I think the reason I chose this title is because it says the nuances I want to convey in the talk today. Not just fat, exploring octadecanoids as novel bioactive lipids in inflammation. And that, I've worked in these compounds for a couple of decades at this point, and a very common reception I've gotten within the field from senior leading experts within lipids has often been, oh, octadecanoids, that's just oxidized fat, that's not interesting, blah, blah, blah. Well, the point of the talk today for me is to convince all of you that, well, it is interesting and that these are important compounds. So with that introduction, I'll go ahead and get started. As Maria said, our sponsor today is Avanti. I'd like to thank them for sponsoring the webinars. So if we look at the role of fat in society, we can see that, as shown here, it's been evolving over time, and our relationship to what constitutes healthy and non-healthy dietary fat is in flux. And there's a lot of unclarity, and many people are unclear. What's good fat, what's bad fat, how much fat should you eat, and so on. And there's a lot of reasons for this. It's multifactorial, but one of the things that I feel is often neglected in these discussions is the acknowledgement that oftentimes fat exerts function via the downstream lipid mediators that are produced from the initial fatty acids. The most classically known are, of course, the eicosanoids that are produced from arachidonic acid, and more recently the docosanoids. But the most common fatty acids, based upon percentage in our diet, as well as the basic basis for all of fatty acid metabolism, is the linoleic acid, ceatine, and its omega-3 analog alpha-linolenic acid. And these are the ones that get converted to the octadecanoids. But there's oftentimes no discussion of the fact that all of our dietary fat gets converted to these potentially bioactive compounds. These are, as a group or compounds, these are called oxylipins, which is a term that I feel is slowly starting to catch on now. It was initially coined by Max Hamburg a little over 20 years ago in this paper that you see here. Down here in the lower left is the definition, which I show again here. I'm not going to read it, but this was Matz's attempt to coin a term that would describe sort of the greater space of oxygenated fatty acids. So of course, this includes the eicosanoids. It also includes the octadecanoids, as well as many other of these analogous compounds. And so it's a useful term to talk about things more broadly than just eicosanoids. Of course, eicosanoids are the most famous members of the class. It's difficult to overstate the importance of an eicosanoid. There's been 2 Nobel Prizes awarded in the eicosanoid space. So one of the problems in working in eicosanoids of the Karolinska is they're sort of like, well, we got the Nobel Prize 40 years ago. Like, what's left to be done? What are you going to do now? Which is actually a little bit challenging with the funding agencies. But what is very clear is that there's extremely important biologies associated with these compounds, and that many of the common processes or common therapeutics that we take for managing pain and inflammation, they target the eicosanoid cascade, as shown here. We see this nice overview of the eicosanoid cascade, and at first pass, this looks very complicated, of which it is. But what I always like to stress when I show this is the simplified version, and that there's actually as many additional compounds not shown here, as well as, of course, the metabolic products of this. So it's a huge molecular space. In addition, there are the omega-3 analogs, which have gotten a little bit more attention recently, piloted by Charlie Sirhan and his group, looking at the DHA and EPA analogs, forming these specialized pro-resolving mediators, or SPMs. But that's not where I want to focus today. I'd like to point out that we see decades of research into these other compounds, but if you look at linoleic acid, this is actually the most common fatty acid in a Western diet. If you think of just total fat consumption that we're eating, it's much greater than these other fatty acids. And that over the last century, we've seen a dramatic shift in a Western diet as we switched our primary fat source from saturated to unsaturated fats. And so, as you can see in the numbers here, 5% increase in our daily energy intake from linoleic acid, which is percentage-wise, that's a huge shift in the amount of fatty acids being consumed. And there's been, as you would imagine, many, many studies looking at the health associations of this rather dramatic shift in the basic Western diet. And a number of the studies of course show interactions with inflammation and metabolic disease and respiratory disease. But the health associations of the effects are really varied. And I could write you a literature review saying that linoleic acid is good for you, and I can write you one saying linoleic acid is bad for you. There's plenty of studies and meta-studies with each point, even within respiratory disease. Some say good, some say bad. It's convoluted. And why is that? Well, I propose that it's because, at least partially, the fact that many times we're looking at the wrong endpoint. We need to focus on these downstream metabolic products of linoleic acid in this case. Understand the ability to convert the primary substrates to their products that exert biological function. And a increased understanding of that, I think, will help sort out some of this confusion we're seeing in the literature about health effects of these dietary shifts. Now, the classical definition of the octadecanoid pathway is shown here. I would be remiss if I didn't at least mention this. We didn't invent the term octadecanoid. It's been around, of course, a very long time in the plant community, and it refers to the canonical formation of jasmonic acid from alpha-linolenic acid. This is the octadecanoid pathway from the plant world. And we're just broadening the definition to include all C18 containing fatty acids. So alpha-linolenic acid can be converted in the same processes as arachidonic acid to give you this alpha-linolenic acid cascade shown here, we're getting, the analogous compounds to arachidonic acid being produced, and of course from linoleic acid as well. So we have these large cascades, but I would venture to say that most people are not familiar with most of these compounds. I assume the screen is too small, you can't see all the individual structures, but there's a lot of compounds, and that majority of the lipid mediator studies to date really focus on a handful, really 5 to 10 of these maximum. There's also, of course, non-enzymatic pathways. So we have the analogs of the isoprostanes or the neuroprostanes from the C18s. And then, of course, there's a whole range of C18 fatty acid substrates. I've talked about linoleic and linolenic, but there's a multitude of different C18 fatty acids. We want to get really crazy. Conjugated linoleic acid is 28 different isomers. So you can start to think of the number of potential C18 octadecanoid products of all these different fatty acid substrates. It starts to be a little bit factorial at this point, and there's hundreds of thousands of compounds in just the C18 pathway. So a little bit of nomenclature before we delve into this. The octadecanoid nomenclature is the same as the icosanoids. Nothing new, nothing we've invented, but just there's been a bit of sloppiness in the field, and people don't always adhere to the rules established for icosanoids. So it's important to push on this a little bit. And so, for example, linoleic acid, the monohydroxys are the HODEs, their keto equivalents are the CODEs, the epoxides are the EPOMs, the epoxides, octadecanoic methylenoic acid, and then we have the dihomes, or the vicinal diols of the epoxides, and then the trihomes, or the triols. And this is the consistent nomenclature that we use across all of the octadecanoids. So hopefully I've convinced you that the structure complexity is immense, and there's a lot of work that can be done here. Primarily the literature to date focuses really on this 9 and 13 HODE from linoleic acid. Why is that? It's because one, the standard companies, they sell these, and they're relatively high abundant, and they're the most common oxidation product of linoleic acid, so they're readily detected. But the vast majority of octadecanoids haven't been studied to date. Why is that? I think a few different points I highlight here. There's a general lack of standardization in the field, or even recognition of octadecanoids as a subgroup of lipid mediators to be studied, and there's really very few chemical standards. And a lot of the work that we've been doing has been just to develop the necessary chemical standards to pursue the studies that we want to do, which leads us to a lack of organic chemists and domain expertise. We've been very lucky to work with some really talented synthetic chemists, but without the organic chemists, none of this would be possible. At some point, people need to make the molecules. And we're finding now that we've started to make them is that happily some of the lipid standard companies are starting to step up there and make some of these compounds as well. But we spent a number of years working on these compounds, and during the talk today, I'll give you a little bit of history of what we've been doing in the field and really why we're in the field. But data from my group and a number of others are starting to show from a number of high impact papers that these are potent lipid mediators of biological activity. But one point I really want to make, and it's these numbers here, and this is, I think, one of the important take-home messages from today, is that we talk about the potency of these specialized pro-resolving mediators, SPMs, are so amazingly potent. But if you look at the endogenous concentration, and of course this is very individual and diet specific, but it's an average in a Western population, we see that it's roughly 0.03% of these EPA, DHA, omega-3 fish oils. Arachidonic acid is roughly twice as much. And well, ALA is over an order of magnitude higher, and then linoleic acid is 7%, so another order of magnitude higher. So even if linoleic acid is a couple orders of magnitude less potent than the omega-3 analogs, the higher endogenous concentration can easily balance that out. So given that this is the major source of our fat, the endogenous concentrations of these are going to generally be much higher, and so the ability to exert effect would be accordingly be greater. So if you look at just a selection of papers that have come out, this is not a systemic review, but we see things involved in metabolism, and particularly in brown adipose tissue, pain, itching, allergy, respiratory disease, particularly immune tolerance and asthma, cardiovascular disease, microbial remodeling, a whole range of different physiologies associated with octadecanoids in journals that we've all heard of that are well-respected journals, showing that octadecanoids like the dihome are actually lipokines, hormones with the potent functions at low concentrations. So hopefully I've convinced you that octadecanoids are exciting compounds, important compounds, and they're worth studying. So now I will switch gears a little bit and show you what we're doing with them and how we've gotten involved with them. As Maria said in the beginning of the talk, my lab focuses on respiratory disease, particularly asthma and COPD. So I will give you examples today from, well, asthma and COPD. When I talk about asthma, I like to point out that it's one of the major non-communicable diseases in the world. And it's a chronic disease of the lungs. And if you look at the global incidence, it's well over 250 million cases worldwide. So it has a major impact on morbidity as well as mortality. And it's one of the most common diseases among children. One of the challenges in working with it is that it's a heterogeneous syndrome. And I won't go into this today, but just to raise the interesting point, it's not the sense that you don't have the asthma. You have a series of symptoms that your respiratory physician will diagnose as asthma with specific presentation of symptoms that require specific treatment. So it's a very good disease for precision medicine approaches to treatment, just because there are so many personalized subtypes. But that's another talk. So when we're working with respiratory disease, when we can, we really like to work as close to the organ of the disease as possible. Of course, in this case, the lungs. So if we actually can get samples from the lung, that's fantastic. So we particularly work with something called bronchial lavage fluid or BALF fluid or BALF. This is collected when the patient, shown here, undergoes a procedure called a bronchoscopy. But basically, they put phosphate buffered saline into the lung, they recover this, and this solution here is what we can analyze. I love to show this picture here. This is the BALF fluid from a non-smoker versus a smoker. So we've washed all of that lovely tar layer off the epithelial lining of the lung with the PBS. So if you smoke, this is what the inside of your lung will look like. So this BALF fluid is very nice to work with. It's, like I said, it's essentially PBS with a little bit of protein in it after you recover it, and it's quite straightforward to quantify our favorite lipid mediators in these solutions. So we take the BALF fluid, we apply, in this case, this is our older method for just, we call it a general oxylipid method, which is developed primarily containing the icosanoids with a few of the octadecanoids thrown in, just because the standards happen to be available. And as you'll see in the talk today, it's because the serendipitous nature of available commercial standards, we included them, and it led to all this discovery that would not have been possible if we didn't have the standards. If you want the details of these methods, they've been published a while ago. I have the citations, you can look those up. So we take this method, and I'm going to show you a couple different ways we've applied this looking at BALF fluid. This is a study we did a number of years ago where we looked at asthmatics, and the study design was interesting in the sense we had healthy controls, then we had a healthy population of asthmatic controls, and we provoked these poor asthmatics with a birch allergen challenge. So they had a known allergy to birch, and so we provoked them with birch pollen to get a response in them. Then they underwent a bronchoscopy, we collected the fluid, and then we analyzed it by mass spec, looking at our favorite oxylipins. The results are shown here. So these types of plots I'll show a few times today, so I'd like to talk about them for just a minute. So this is an analysis called OPLS, or orthogonal projections of latent structures. It's fairly common in the metabolomics field. It's a supervised multivariate modeling approach. And so each of the the circles here, this is an individual in the study, and includes, in this case, all of the lipid variables in the supervised model. And then you get some statistical metrics for the quality of the model. So in this case, the model is fairly decent, but it's overfit given the large gap between the R squared and the Q squared. But what we can see in the top model here is we're able to pull apart in the scores plot the healthy controls versus the asthmatic controls fairly well. And then here we have the healthy controls versus the asthmatics following the provocation. And we get an improved model, improved separation. We look at which lipids are driving that. We look here in the loadings, and I don't expect you to read all the different lipids, but if you see what's driving it, everything in blue, for example, these are lipids that are produced from an enzyme called the 15-lipoxygenase. So what we can see here is that, okay, these are lipids that are associated with asthma. And this has been known before that 15-LOX produces octadecanoids and other mediators involved in asthma. But what's more interesting, since we want to look at the effect of the birch allergen provocation, particularly with the study design, we want to know what's happening with that, is we can make something called a SUS plot, which is shared and unique structures. Basically, it's a correlation plot. We're correlating the two OPLS models. So you look to see which lipids contribute to one model versus the other model. And everything, of course, in the diagonal, it equally contributes to both models. For example, all of our 15-lipoxygenase lipids that we just looked at, they're essentially on the diagonal. So these are associated with asthmatics, whether you're a control asthmatic or a provoked asthmatic. This is lipids that are reflective of the disease, not necessarily in response to the birch allergen provocation. Where we look at these lipids over here shown in the gold circle, which are closer to the allergen provocation model. So these are more specific for the response to the birch allergen provocation. And these lipids are the ones you see here. So these are epomes and dihomes. Hopefully these acronyms are a little bit familiar now. These are the epoxides and diols of linoleic acid. We look here, here's the structure. So linoleic acid is converted by cytochrome P450 to the epoxides, which are then opened up by the soluble epoxide hydrolase to the corresponding vicinal diols, epomes and dihomes. So what we're seeing here is that these octadecanoid lipid mediators are actually markers of pulmonary damage associated with the birch allergen provocation in these individuals. Another example shown here, these are healthy individuals who are exposed to diesel exhaust or filtered air, and then they looked at the levels of these different oxylipins in the lavage fluid again, and levels of this linoleic acid dihome are increased in individuals exposed to diesel exhaust. A study that I really like to design that there's a unique study we did a number of years ago now, was we brought in individuals to the lab and we put them on an exercise bike, and they had this bike on this for 2 hours. And then we did the same thing on a subway stop in central Stockholm. So basically on the platform, they sat there on the bike for 2 hours, just slow pedaling, not fast, just slow, just to control for mouth breathing, but just breathing subway air for 2 hours. So we did this in subway air and filtered air with asthmatics and healthy controls. And then 24 hours later, they underwent a bronchoscopy. You looked at the lavage fluid, same methodology I've talked about. And then what you see is here. So it's a plot of the ratio of the subway air to the control air, the filtered air, and then red is asthmatics and black is healthy. And what we see is all these lipids are actually increased following exposure in subway air in our healthy individuals. And these are all products of linoleic acid, except for this one DHA product. So all of these are octadecanoids. And we're a little confused at first because the hypothesis going in was that, okay, asthmatics, they have weaker lungs due to their illness. So we would expect that we'd see higher concentrations of these lipids or a bigger response. But we see the opposite. We see that the healthy individuals have the greater response and that the levels increase with them following the quote unquote toxic exposure to subway air. And the interpretation we came up with is that if you are a healthy individual, your lung responds well or has a strong response to a toxicant exposure. So breathe in the subway air, and your lung says, oh my God, what is this horrible stuff? And you initiate an inflammatory cascade and you produce these compounds. Whereas the asthmatic lung is already compromised, so it's unable to mount this pro-inflammatory response to the extent a healthy individual can. But for the purposes of this talk, what's an interesting component is the fact that, again, it's all the octadecanoids that were the main mediators responding following the subway air exposure. Moving over to an example from COPD, this is a study called the Karolinska Cosmic study, which was designed to look at the effects of gender and smoking in the onset of COPD. The study design is as follows. We had 50-50 men and women. We had 3 groups. We have our healthy never smokers, our current smokers. We call them healthy smokers, but there's no such thing. So it's really smokers with normal lung function, and then COPD smokers. And then we had a couple COPD ex-smokers included in here as well. You'd be surprised how hard it is to find COPD ex-smokers, even with a diagnosis they keep smoking. But this was the study design. We collected ball fluid from these individuals, applied the same methodology I've been talking about to these samples. We quantified 87 oxylipids in the ball fluid. Here is the initial PCA plot and decent model. We're able to pull apart our non-smokers from our smokers. We see a sort of a disease trajectory from our healthy non-smokers down to our COPD smokers. And so there's a lot of structure. We're quite happy with this. So we went to the OPLS model again, this orthogonal projection to latent structures. And we're comparing in green the COPD smokers from red, the smokers with normal lung function. If you look at the model, it's not particularly impressive. The numbers are relatively low for the Q squared and R squared, but it is significant. But this model includes both genders. And remember I said a key driver of the cosmic study is to look at the role of gender in COPD. So we performed a gender stratification where we only looked at males separately or females separately. And here is the female only model. And importantly, there was no model for the males. There wasn't enough structure in the data to make a significant model. For the females, R squared has gone up, or they've both gone up, but we see that the gap between R squared and Q squared is fairly high. A general rule of thumb we have, if it's greater than 0.3, it suggests the model is overfit. So we decided to do a round of variable selection. Following that, we made this model where we again stratified by gender. And again, we show you the female model because there was no corresponding male model. Now R squared and Q squared are very close. They've gone down in value, but still they're fairly decent and they're quite close. We had a good p-value. So we said this is our final model. So this is the scores plot. What's driving it? I'll go here at the loadings and we can see here are the 9 lipids that drive this. So what are they? I assume you don't remember all the acronyms. So everything in red, these again are octadecanoids shown here. And then in blue are the arachidonic acid products of thromboxane synthase. But let's focus here on these top 5 in red from linoleic acid. Now we thought, this is lovely, but is it real? It's still a relatively small study. We'd really like to validate it. So we were lucky enough to get our hands on a validation cohort. So in this case, we had a different group of patients that were recruited from a different hospital, brought into the lab, and they were analyzed on a different mass spec by a different postdoc. And then we took the first model and we used it to classify the individuals from the second cohort. And we had 93% accuracy, or 92%. So we were quite happy. We said, okay, we've really validated it. We believe this. So let's go back and look at these top lipids again and see what they mean. Well, the epomes and dihomes, we've seen this figure before, epoxides and the diols. And then going back in the ancient literature, this paper was published by my PhD advisor the year after I joined the lab. I remember very well when it came out. Nature Medicine paper is a big deal. And it's just kind of funny that here I am 25 years later giving a seminar talking about this paper. Sometimes you do learn something during your PhD. What was interesting in this paper was they reported that the leukotoxins of these epoxides or epomes were associated with something called acute respiratory distress syndrome, or ARDS. Now, 2 or 3 years ago, nobody had heard of ARDS and no one really cared about it. Now with COVID, suddenly ARDS has become much more of interest and a much more common syndrome, unfortunately. And this is what's associated with severe COVID and what's responsible for a lot of the severe COVID associated mortality. They also reported that the diol, the dihome metabolite, was toxic to pulmonary alveolar epithelial cells. And if you inhibited the soluble epoxide hydrolase, you reduce the epoxide-associated toxicity. So the epoxides are essentially a protoxicant, which you can inhibit by ablating the epoxide hydrolase. And GSK actually has been working on developing inhibitors of the epoxide hydrolase for treatment of COPD. If you go back even further in literature, now even a decade before, now we're in the 80s, Japanese groups, Ozawa and colleagues, published a number of papers on leukotoxin, where they showed that what you see here is rat lungs, 10 minutes after you've injected the epoxide, these EPOM, into a rat. You get this massive pulmonary edema and this damage associated with these leukotoxin epoxides and diols. So we're seeing this consistently now from the first studies I showed you with the birch allergen provocation, the asthmatics, we saw increases in the EPOMs and dihomes of linoleic acid. We see this now with COPD and even in these early rat studies. So a very small study that was done looking in COVID, which you're looking at here, it's not a great figure, but this is what was in the paper, is what they're looking at here is the circulatory levels of the epoxides and the diols of linoleic acid in a control population or 6 different severe COVID patients. And they've taken this at different time points. And so you can see that in some individuals, there is an elevation of the circulatory levels of these compounds. Looking at the association between, again, these epoxides and diols with COVID severity. Then we were lucky enough, we were fortunate enough to be involved in an interesting study that was done here at the Karolinska, looking at the potential to treat COVID severity with an infusion of omega-3 fatty acids. And so individuals were brought into the hospital and they were given IV omega-3 for 3 days. And then we received blood from that, and we looked at the circulatory lipid mediator profiles of that. So here's the scores plot showing you in red the placebo or blue the N3 treated group. And we look at the loadings, we see what's driving this. There's a few compounds in here we might recognize. Here we have our dihomes, and there's some other codes and hodes from linoleic acid. But to make it a little bit easier to understand, if we look here, so here's the 910 dihome and 1213 dihome, right? The linoleic acid diols. Here's the placebo baseline, and then the N, 3 days of treatment versus omega-3R. And we see increases with placebo and very clear decreases in association with omega-3 treatment. So omega-3 treatment for 3 days decreases the formation of these toxic linoleic acid diols, which are associated with ARDS and severe pulmonary damage. So it's quite exciting to look at the potential for octadecanoids and what they're exerting in respiratory disease. Now, going back to the COPD model, we talked about the top 5 lipids, but I've really only focused on the top 4. So what's this top one here, this trihome? Well, trihomes are these trihydroxy metabolites of linoleic acid. There's a number of these. You can have the triols in many different positions on the linoleic acid backbone. The 9-10-13 and 9-12-13 are the most studied. Guess why? Because those are the standards that are sold the most. And if you look at them, given that they're triols, there are 3 different chiral centers. So we have 2 to the 3 or 8 different isomers for each of the different structural isomers. So for these 2 that are the most common ones, there's 16 different isomers, which look like this. So essentially all studies to date looking at trihomes haven't done a chiral analysis on this. So they just generally report a total trihome signature for something that actually is 8 different molecules. So given that this was the #1 driver for looking at the transition from a healthy smoker to a COPD smoker, we got very interested in understanding how this compound is being formed. So we had a very talented postdoc, David Fuchs, who worked with Mats Hamburg again to develop a method to quantify all the different isomers, each individual enantiomer of the different trihomes. This is what you see here. This is published a few years ago now. And for each of these, you can see we get our 8 beautiful peaks. But I would like to draw your attention to the retention time on the x-axis. This was a very, very long method, and it was 107 minutes per run. So not exactly for looking at large cohorts with really wide peaks, but it worked at least. So we took this method, and also we had an standard LC reverse phase method where we could look at the pairs, but there's no chiral separation here. So the enantiomeric pairs are not separated. So we did this first because this is just faster than that awful chiral method. And for all the pairs, we could see that in all 8 pairs, the levels increased in the ball fluid in the transition from a healthy smoker to a COPD smoker across the board. So then we looked at the chiral data, and that's what you see here. And here I need to explain something called enantiomeric excess, or EE. This is a way to look at the enrichment for one in a chiral center for one isomer or the other, in this case, R versus S. The reason why this is important, looking at the 13 position, because if this is hydroxylation is formed via the enzyme 15-lipoxygenase, then that will give you exclusively the S isomer. Where if it's formed by autooxidation, it will give you racemic 50-50 RNS. So by looking at the enantiomeric excess at the 13 position, we can then infer the synthetic source of the compound. How is it being formed in COPD? So if you look in sort of red and maroon here, this is our healthy smokers, and then in green is our COPD smokers. So the EE is higher in the healthy smokers than the COPD smokers. Now let's unpack this for a minute. So we saw in the last slide that the levels of the compounds, they all increased in the transition from healthy to COPD smoker. Levels went up, but the EE goes down. So what that tells us is that the sort of baseline levels in a healthy smoker, they are coming from, at least partially, from 15-lipoxygenase activity, most likely via the diet, to be honest, but enzyme synthesized 13S. But then when you increase the levels, when you transition from healthy smoker to COPD smoker, that process is actually autooxidative. That's a racemic. And so we're seeing the EE go down. So we had a much clearer understanding of the mechanism, how these things are being formed. And this is very consistent with what's known about the gender-based differences in COPD. It's been a number of papers showing that actually the oxidative defensive mechanisms are downregulated in women relative to men, which is part of the reason they're more susceptible to the toxic effects of smoking and have increased COPD rates. So this was very consistent with what was known about it, and interesting that we could actually disentangle the mechanisms. Then we got curious to know which different cell types could be making these trihomes. And so we looked at a number of different immune cell populations. We looked at eosinophils, neutrophils, and platelets. So what you're seeing here is that these are the data for the eosinophils. So we take the eosinophils, we isolate them, incubate them with linoleic acid, and then sure enough, we could detect all of our different isomers. But we did not detect any trihome product with neutrophils or platelets. So in our hands, this was solely coming from the eosinophils. If you look at, again, the enantiomeric excess following eosinophil-based synthesis, you can see it's very, very high, in some cases over 80%. So this is clearly a 15-lipoxygenase mediated process. But just to be sure, we want to do the pharmacological study, and we actually incubated eosinophils with a 15-lipoxygenase inhibitor. We can show very clearly we could ablate production of trihomes, whereas addition of either an epoxide hydrolase inhibitor or a cytochrome P450 inhibitor had no effect upon trihome formation. So it's clearly a 15-LOX product. So we proposed this mechanism for a trihome synthesis via 15-LOX, which we published a couple of years ago. So just to make it fun though, biology is, it always wants to keep you guessing. We did that last experiment that we added, and we looked at a cell line, a mass cell line called LAD2 cells. And if you focus here at the 13 position, these guys, they make the R isomer. So we see, the enantiomeric excess is not quite as high as we saw in eosinophils, but nonetheless, we do see that the R isomer is being made. So clearly there's a different route of formation here, most likely via cytochrome P450. But it's just interesting that different cell types have different enzymatic machinery for making these trihomes. So just very quickly, we have a study we've been doing with a collaborator, Phil Hansbrough, down in Newcastle in Australia, and he has a model a murine model for COPD, where these poor mice, they basically are smoked for 8 to 12 weeks. And then we're able to look at the onset of COPD in these mice. And the advantage of doing this, of course, is we can do dietary modulation. So given the associations we're looking at here between dietary fat and disease, we can change the composition of their fat and look how that affects disease presentation. So we've been doing a number of studies with him. I'm not going to give you all the data today. That'll have to be in the talk. But just to show you the type of thing we can do, is that here we just went ahead and we used different concentrations of a soluble epoxide hydrolase inhibitor in the drinking water alongside smoke, and we can see that it affects the number of macrophages that we observe in the ball fluid. So, and also it's a biphasic response, which is quite interesting. We also could show that administering a soluble epoxide hydrolase inhibitor reduced the alveolar diameter, which is an emphysema phenotype. So basically the smaller your alveoli, the less the surface area, so the less gas exchange there is. So that's what emphysema essentially is. And so administering an SEH inhibitor reduced that emphysema phenotype. So looking at the ball fluid in these mice, we can show that giving them dietary linoleic acid increase the levels of the epoxides, the diols, and the trihomes in the ball fluid very nicely. And then if you gave them an insoluble epoxide hydrolase inhibitor, as you would expect, you decreased, you increase the epoxides and decrease the diols and didn't affect the trihomes. Makes perfect mechanistic sense, is based on everything we've talked about. But just to show this in vivo, so we give them the inhibitor in the drinking water, and we can see very nicely in vivo the effects. So I just looked at the time, so we should be moving on a little bit. So hopefully I've convinced you that lipid mediators are more than traditional eicosanoids. So what next? Well, we've been working on developing targeted methods for eicosanoids to basically develop analytical technologies to detect these compounds more broadly. When we wanted to develop the method, we wanted something able to discriminate chiral species, to be good for large-scale studies, to be sensitive, and offer relatively broad compound coverage. So happy to say that very talented postdoc, Alessandro Quaranta, has spent the last couple of years working in this space and has developed this very nice method where we can look at 103 different octadecanoids using supercritical fluid chromatography with a Waters UPC squared system. So we have here the different compounds shown here from the different fatty acid substrates. We cover a range of different enzymatic and non-enzymatic processes. The SFC separation is just, it's really, really nice. We've been particularly happy with it. If you look at the trihomes, this is a good example since the trihomes gave us so much trouble before. Remember, we have 8 isomers for one specific trihome. So if you look by typical reverse phase LC separation, if you're lucky, you can get 4 peaks like this. Usually it's 1 blob to 3 peaks, but this would be an ideal situation. And then for us, I showed you earlier that we had this chiral method, which got all 8 separated, but took over 107 minutes. Now, by SFC, in less than 12, we can separate out all 8 of these. So this was just really beautiful by SFC. But not everyone has an SFC, and we want to make octadecanoids available for everyone. So we also developed an LC separation method that works really quite nice as well, but you just don't get the chiral information. But if you don't want to do chiral chromatography or don't have an SFC, then this LC method will work just fine. This shows you the precision of the different methods for a few of the compounds, and as you'd expect, the 2 methods correlated fairly nicely. Quite happy with that. Here are the analytical specs. I'm not going to read this slide. The paper is in ChemArchive. It's been there for a while now, so if you want to take a look at the details, download the paper. This is all there. The thing I do want to stress, though, is that we had to synthesize 70 different compounds to do this. So roughly half of the SFC and the LC platforms were custom synthesized to do this work. So just the compound availability is really the bottleneck in doing this type of work. So hopefully following this work, well, it's not hopefully, we're already seeing it, that some of the standard lipid companies are already making these compounds, making them more available after seeing our work. So this is really gratifying to see availability increasing. But all this work, was it worth it? Well, if you look at just reference mouse and human plasma, sure enough, these compounds are there. So we're seeing the octadecanoids present in relatively high levels. So it was worth the time to synthesize these things, make the method, because the compounds are there. And now we can start seeing what types of function we can observe. In the last few minutes, just giving you an idea of what we're doing with it, we're very interested in the ability of the gut microbiome to convert these dietary fatty acids to these octadecanoid lipid mediators, particularly looking at the gut-lung axis, since my lab obviously looks at respiratory disease. So to look at these microbial products, again, none of these standards are available. So we have a postdoc who we're very lucky to recruit, an organic chemist, comes to us from the Montpellier group with Jean-Marie Delano and Thierry Durand. And she's been working very hard synthesizing these microbial metabolites. So we have a whole range of standards now that we've been using and functional assays to see what types of chemical or biological activity they have. So I don't have time today, but we've been doing a number of things like phagocytosis and chemotaxis just to understand what type of function is available. To give you just one idea, for example, with a collaborator, Julia Serre-Flambier in Munich, they take mice, they isolate macrophages from the monocyte-derived macrophages from bone marrow, and then they incubate them with LPS, and then we can look at cytokine production. And if you incubate them with one of our octadecanoids from alpha-linolenic acid, you can see a roughly 50% reduction in pro-inflammatory cytokines in this macrophage model. So there's some interesting potential activity with this. In addition, we've been looking at different bacterial cultures and their ability to synthesize these octadecanoids. And so we just take the different cultures. Here's two different cultures we're working with, and we incubate them with fatty acid substrate. And this is just right now, yes, no are the compounds present. So we substitute with LA or alpha-linolenic acid, or this is just the blank. And we can see we can make, a number of these compounds are being made as one would expect. So that's all very exciting for us. So to wrap it up, since time is running out, I hope I've convinced you, based upon the title, that dietary fat is an important source of substrate for the production of lipid mediators. This quote-unquote fat is in fact biologically active. There's hundreds of thousands of novel compounds that have yet to be studied, but the initial studies certainly demonstrate potent biological activities. We saw that linoleic acid-derived mediators, these epomes and dihomes, particularly the dihomes, may be involved in the pathophysiology of COVID-19, given their association with lung injury and ARDS, and that potentially targeting the SCH could be useful in treating the ARDS component of COVID. And that the microbiome demonstrates an ability to synthesize these octadecanoid lipid mediators, and then gives you the potential for dietary modulation for substrates to form these lipids, and potentially for treating different disease states. So lastly, I hope I leave you with that the octadecanoid pathway is an exciting new research area. I think it's really great to see a new area of lipids opening up. There's a strong need for people interested in this who have the necessary tools and interest to explore this. We particularly need good organic chemists, biochemists, analytical chemists, and so I really see this as an opportunity, and I hope that everyone here does as well. I'd like to thank a number of the people who worked on this, particularly Alessandra Quaranta, who developed the octadecanoid methods, Hildur Anadotter, who worked with us in the COVID studies, and Joanna Revaux-Cavalier, who did a lot of the synthesis for the microbial work. Thank you for your attention. And the last point I'll make is that we have a number of openings in the lab. We just got a couple of grants funded, and so we're looking for a postdoctoral researcher and a PhD student. If anyone's interested, please contact me. So with that, I'll conclude and take any questions.
Thank you very much, Craig. Like always, a very amazing talk and very exciting area. So we indeed have questions. So I will read your name and you will be unmuted and you can ask your question. So there is several questions from Konstantin Biryukov. So please. So hello? Konstantin, can you unmute yourself? Let me see if I can do that. Just a second. So maybe then I will just read out the questions. So there are several questions from Konstantin. So the first one, is production of linoleic acid-derived oxylipins somehow controlled and coordinated by the host as a part of host response to insult? Oh, I guess Konstantin unmuted himself.
It's a great question, and the answer is yes and no. I would say, in the sense that definitely some of these are controlled and coordinated by the host in response to, as you say, insult. These linoleic acid is, it's esterified in phospholipids, and it's released by phospholipases to make the substrate available for enzymatic conversion. So activation of those phospholipases is an important part of an inflammatory response. In addition, there's just very large levels of linoleic acid, either free or esterified, and these can be oxidized via enzymatic and non-enzymatic processes, whether it's oxidative stress or any shift in the oxidative environment, like we saw with COPD, or to see some sort of shift in oxidation causing a non-enzymatic formation of these oxylipins.
Konstantin, would you like to ask your following question? I can see you unmuted, but no sound. Okay, then I will continue reading. So probably the second one you partially answered. So are those compounds result of enzymatic or non-enzymatic conversion? And
yeah, I think we answered that one.
Yep. And the last question from Konstantin is: can you identify groups of good and bad oxylipins with respect to their role in asthma and COPD? Again,
Absolutely.
is there any regulation of their balance and health and disease patients?
No, absolutely. I mean, if we broaden it to oxylipins, not just octadecanoids, but yeah, there's a balance of these, and some of these compounds even have, bronchodilatory, bronchoconstrictive effects depending upon when and where they're located. And so there are, like PGE2, prostaglandin E2, it's called a Janus molecule because it has two faces. Sometimes it's beneficial, sometimes it's not. If you looked at the story associated with the omega-3 derived pro-resolving mediators, they would say that these would be, as you put, quote unquote, good oxylipins that would be associated with the resolution of an inflammatory event. So yes, there's sort of a battle between pro-inflammatory and pro-resolving oxylipins involved in this inflammatory cascade.
Thank you. The next question is from Grant Hedge. Could you unmute yourself and ask your question?
Hi there, can you hear me?
Yep.
Yes.
Hi, great. Wonderful talk. I really enjoyed that. So I was curious with the, and I guess you alluded to this a little bit earlier in just that last question about the mechanism of the omega-3 treatment in that trial with COVID-19 patients. Do you think it might involve possibly something like prostaglandin E3 signaling or competition of PGE3 species that are generated from the omega-3 with PGE2 that's generated from the COX pathway for EP receptors?
Great question. Really, thank you, Grant. Absolutely. I mean, the hypothesis going in, to be honest, was more of the canonical SPM pathway. That by giving EPA, we'd see an increase in these resolvin-like compounds, and that's not what we observed. But from a physiological standpoint, clearly there was benefit of the omega-3 treatment. So what is the mechanism then? And I think the PGE3 comment here, that would make a lot of sense to me. One of the things that if I was looking at the data I presented, the question I would ask is that I reported that the linoleic acid dials decreased. You'd say, well, yeah, that's not so surprising. You're putting in gram quantities of omega-3 fatty acids via IV. So isn't it just kinetics? Is it substrate competition that's occurring? And the data that it's not in the slides, is that there was no change in the arachidonic acid diols. So their levels remained constant over the treatment, and only the linoleic acid went down. So that exactly that I found that the most fascinating. So there is something specific about this pathway that's occurring. So the PGE theory is very intriguing, Grant.
Okay, thank you very much. Excellent talk.
Thank you.
Thank you. The next question is from Kourosh Hushmand. Maybe you can ask a question yourself. You can unmute yourself.
Hello, Greg. Thank you for the great talk. Yes, because I am working with long-chain fatty acid and trying to develop a method with LCMS. So I was wondering these octadecanoids, if they are ionizing well with electrospray ionization, or you need to derivatize them prior to the LCMS analysis.
I know that's a good question. The short answer is that they ionize just fine. I mean, a lot of the early work in the caisinoids as well as octadecanoids was done by GC, but with derivatization. But these days they ionize just fine, and most people use LC. So we don't derivatize. The only thing I would say is that sometimes the fragmentation patterns are not so specific. So the MRMs can be a little bit challenging with them. I mean, if you have just a single hydroxylation, you don't get very good fragmentation sometimes. So that's the biggest problem.
Thank you.
Thank you. The next question is from Nancy Duarte. Nancy, would you like to ask a question yourself? You can unmute yourself now. Okay, so Nancy, you cannot unmute probably. Then I will read out the question. So she writes, I found the talk very interesting. I wanted to know if you have evaluated the effect of the medicine consumed by asthmatics like bronchodilators in the production of octadecanoids derivatives. I assume the asthma patient were consuming these medicines, and that may be a confounding factor.
Great question, Nancy. Yes, we always stratify by medicine when we're dealing with these patients. We've published a couple studies recently where we've shown that you see therapeutic specific effects on just metabolites in general, not just octadecanoids or oxylipids, but just we did this with metabolomics as well. And that bronchodilators, we've never observed any effect. With steroids, it's a different story. With icosanoids, we haven't seen any effect, but there are a number of other metabolites that are either depressed or increased with both oral and inhaled corticosteroids. So in our recent paper, we evaluated bronchodilators, OCS, ICS, theophylline, anticholinergics, and everything is compound specific and medicine specific. But the point that you raise here is that, as you say, it's a confounding factor because you're never going to get a population of asthmatic patients that are not taking their bronchodilators. So how do you control for that? Some of these things get a little bit difficult to work with. So you can try and do, how many times do they use their bronchodilator, but these are patient reported, it also gets difficult. But anyway, I'm answering too long, but it's an important question and it needs to be included in the analysis.
Thank you. And then we will take a last question or two questions from Nazak Naureddin. Could you unmute yourself? Yep.
Hello, yes, can you hear me?
Yep.
Thank you. Thank you so much for a very interesting talk. I have two questions. My first question is actually back to the COVID study where they supplemented with omega-3. Was ALA part of the treatment, or was it just EPA and DHA?
EPA and
because
DHA.
There is
no ALA.
Okay, so no ALA. Because there's a study already published showing that supplementation with ALA increases certain oxylipids derived from linoleic acid in a mechanism that is not dependent on the SPMs. So I wanted to know if ALA was part of the treatment. And my second question is, you showed several maps of all possible oxylipins derived from ALA. Is there like a database or maybe a recent publication you would recommend where we can get an up-to-date map of all possible oxylipins? Thank you.
Unfortunately, there is not. The maps I shown are just brute force made maps by poor Alessandro, just in PowerPoint and ChemDraw, putting that together.
Okay.
So it's very custom, and it's certainly not quote unquote all octadecanoids. I mean, there are many more. It's all the ones that we have included in those maps. I've been trying to convince people like Cayman to make an octadecanoid chart, like their isonide chart. I think it would be useful for the field.
Yes.
Because we need to standardize this so we know what are the pathways, what are the compounds, then we can study them. But right now, without that, it's very unclear what we're looking at.
Exactly. Thank you. I thought they perhaps were from Cayman because last time I checked, there wasn't any. So, but anyways, thank you for the
nope.
answer.
If you're on the call, Cayman, you can hear that. People want the charts.
That sounds like a task for lipid maps, EpiLipidNet, and Wiki Pathways to do. So that's something which can be definitely done, and it's quite an interesting point. Thank you very much, Craig. So we are at the end of our time for the webinar.
My pleasure.
Thank you very much, and thank you all of you who attended today. Bye-bye.
Thank you so much. Bye, everyone.